Good afternoon, everyone. Today we have Palak Trivedi. Palak is, well, he's a medical doctor, but with a very strong background in as a PhD in immunology, right? He was, you did the PhD in immunology, and then now you are a medical doctor, MD, PhD. You have 50% of your time is on research. And Palak has been now focused on well, on gut-liver axis and primary sclerosing cholangitis, and now you're going to talk a little bit about the gut mucosal immunity and in DILI, in drug-induced liver injury. So I think today is a very interesting and very exciting seminar. And Palak, we are here ready to hear you.
Yeah.
So go ahead whenever you want.
Oh, right. Sorry. Just making sure. Let me just try that again because it just stopped sharing.
Okay.
Let me just give you 1 minute to reopen the slides. And then. Okay, you can see the slides yet?
Yes. Perfectly.
Good. Yeah, sorry, just cut out then. Now, thank you very much for this kind invitation, and apologies again for my tardiness this afternoon, just traveling from one part to another. So so today I'm just in the next sort of half an hour or so, I'm going to talk a little bit about the potential role of gut mucosal immune responses in mediating drug-induced liver injury, both in terms of augmenting risk as well as attenuating risk, and trying to use particular examples, okay, just based on the literature. So 1 second, my slides keep on freezing every time I try to move them forward. There we go. So these are my disclosures. So I mean, to an audience like this, I don't need to go through this, the central principles regarding, gut-liver immunity. The whole theme of the gut-liver axis, is very well studied across all elements that I'm interested in, such as chronic cholestatic injury and immune-mediated liver diseases, elements such as non-alcoholic fatty liver disease, alcohol-related liver injury, and also sort of in chronic liver disease. And the core pillars which people tend to focus on, which again will form some of the cornerstones of my talk in the next few slides, will be around the potential role of the gut microbiome, dysbiosis, but also around disrupted bile acid pathways and how they may be modulating the impact of particular drugs that cause liver injury, but also that cause idiosyncratic injury elsewhere in the GI or hepatobiliary tract. And also, what can we learn about dysregulated mucosal immune responses from the immune-mediated liver diseases that perhaps need to be studied in further detail in the drug-induced liver injury field. So this is a disclaimer here. When we're trying to study gut microbiota, we have to accept some convenient versus inconvenient truths. Almost all diseases, be they liver-related or even in the mental health field, even in the respiratory field, report some element of dysbiosis in inverted commas. What that means is that the diversity of the gut microbiome is different compared to what we would associate with health. Secondly, there is a slight abundance of some populations and more abundance of others, coupled with a reduced abundance of what we perceive as investigators to be more protective species. Of course, there's going to be a literary bias where people tend to favor more positive results, which I will come on to shortly. One of the things that is quite reproducible, at least in animal models, is transferring of disease, wherein we can transfer gut microbiota from pathological models, for example, in sclerosing cholangitis and fatty liver disease, to wild-type animals, and that is associated with transfer of disease. What is less convincing, though, is at least in chronic diseases, with the exception of IBD and chronic diarrhea, transferring healthy stool, healthy microbiota from, as I say, from a wild-type mouse to a disease mouse, attenuating liver injury. That is much less reproducible in itself, and that is a lot harder, a lot more difficult of a bar to sort of to reach. And when people tend to study intestinal dysbiosis, whether it be in drug-induced liver injury, autoimmunity, there is a tendency to work in silos. There are very few papers which will look at a NAFLD group in comparison to a PSE group from the same geographical population. But within an individual population of patients with ALD, with NAFLD, with hepatic encephalopathy, we start to see some differences compared to the healthy state. The inconvenient truths therefore lead on. There are very few head-to-head comparisons. There are less studies comparing acute versus chronic liver injuries of the same etiology. For example, acute versus chronic drug-induced liver injury, which does occur. And when we look at studies of the gut microbiota, those of stool microbiota, at least in man, predominate rather than those of mucosal adherent gut bacteria. And that is fraught with some confounding factors. Stool does not represent the colonic microbiome. It represents almost quite a lot of the small bowel, the large bowel, and of course contaminants by skin flora. So that is one reservation. When we talk about microbiome, of course, there are very few studies that look at the link with diet, with age, and with geography. I think some colleagues are on the call. I examined an excellent PhD thesis this afternoon, up in Nottingham, and it was nice to see some of the studies looking at the impact of diet alongside changes with the gut microbiome, but those are very few and far between. Now, with those caveats in mind, let's begin. When we look at the distribution of the gut microbiome throughout the GI tract, it's obviously very clear that we are more microbe, more prokaryote than we are eukaryote. We are more bacteria, we have more bacterial cells than we have human cells within the human body. And that, of course, differs in composition throughout the GI tract. But before we get into the principles of in the meat of the talk, what have we learned over the last few years as to what are the factors that impact the gut microbiome and, of course, impact how the gut microbiome interacts with certain drugs, food antigens, and the like? Well, there was this landmark paper published by Rins Wiersma's group in the Netherlands last year, and the Dutch Microbiome Project looked at several thousand individuals and families, collecting a wealth of very rich demographic and phenotypic data. The N numbers you see around this plot are actually the number of variables relating to that particular domain. So, for example, exposomal variables, there were 10. Dietary variables, there were 21, and so on and so forth. So this is an incredibly rich resource, which has really taught us about the factors impacting what composes the gut microbiome. Now, one of the key take-home messages here are that the genetic background, whilst contributory, is probably only explaining about 7% or less than 7% of what makes up our gut microbiome. Heritability is a very small proportion of what composes our gut microbiome. And it was about, I can't remember the exact statistic, it was about 40 to 45% or on average of the gut microbiome is contributed by the environment and, in fact, by cohabitation. In these violin plots, what you can see from left to right is a series of plots which tell us how different between the different pairings the gut microbiome is. The lower the median, the more similar the gut microbiome is in each of the individual pairs. So if you look at people who cohabitate on the left-hand side, those are people who live together. So, and the control group here is the yellow, the non-cohabitating pairs. Partners, this is where their median lies. Parent and child who live together have a more similar gut microbiome than partners, so couples who live together, and siblings have the closest microbiome to one another when they live together as compared to parent and child and compared to partners. When you look at non-cohabitating pairs, you can see that there is really no difference, suggesting that if you're siblings or your mother and child, father and child, that your gut microbiomes are going to be as different to one another if you're living apart as they are if you are not blood related. But look at the far right here. When you look at people cohabitating, first degree relatives cohabitating, they have a much more similar gut microbiome, suggesting that in this panel, compared to the middle panel, living together is one of the factors that that's associated with what the gut microbiome is, and that becomes relevant when we start to learn about transferring of liver disease risk and how the environment may play a greater role than genetics. So, as I mentioned at the start, changes in gut microbial composition are illustrated across many different health states. This is a very old slide. I mean, if you were to redo this, we would need about 10 more slides, but this is just illustrating some of the changes in the gut microbiota across several disease health states. And how does the gut microbiota start to impact the risks of developing drug-induced liver injury and other forms of liver injury that share similar phenotypic features? Well, what you can see here in this particular infographic, now, we are fully aware that all the factors on right at the very top, these exposomal risks will impact not just the composition of our gut microbiome, but how our gut microbiome behaves. And in particular, what we see in states of health is an increased diversity, increased proportion of symbionts, particularly generation of protective metabolites like butyrate and short-chain fatty acid producing species, and increased secondary bile acids like isolithocolic acid and oxolithocolic acid, which, as I'll come to show you, help polarize intestinal mucosal immune responses towards more regulatory T cells rather than Th17 and IL-17 secretor cell types. And yes, they foster tolerogenic phenotypes in health. And the reverse is true when there are states of disease. Now, these are the 5 main phyla that we're going to talk about and how they may impact disease. And in chronic diseases, be that acute progressing to chronic liver injury from medication, or indeed drug-induced autoimmune-like syndromes, the richness of the bacteria, the richness of the whole gut microbiota pool tends to go down, and we get increased expansion of pathobionts. And in PSE, which I'm not going to talk about in any depth here, there were several nice animal models published about 4 years ago showing that you can induce PSC by transferring the stool of humans who've got the disease to notobiotic mice, simply by an adoptive transfer of organisms, and that was predominantly driven by the pathobiont Enterococcus. So how do certain drugs affect the gut microbiome? Now, I'm not expecting anybody to read and digest this slide. It's just simply to show from this very nice and thoughtful review published in Gut a few years ago that commonly used drugs, even some that you can get over the counter in the UK, we can get proton pump inhibitors over the counter now, they are very critically affect the composition of the gut bacteria. And I use that word critical not lightly, because as I'll come to show you, this has long-term pathogenic consequences. So when we have commonly used medications, we ask ourselves 2 broad questions. First of all, what is the effect of the gut microbiota on the drug? What particular if you have a particular drug on the y-axis here, how many bacterial strains are going to be responsible for metabolizing that drug? That's what's shown on the y-axis here. And you can see some commonly used drugs right at the top of this graph. And you can see here that a very high proportion of strains will be responsible for metabolizing those drugs. And on the x-axis, you can see the number of bacterial strains whose growth rate is affected by medication. So the reverse, if you give a particular medication, how many bacterial strains are going to be affected by that particular medicine. So you can see here on the y-axis, things like fibric acid derivatives, antidepressants, they affect a broader range of gut microbial species than drugs right at the very bottom, and vice versa. Now, this is irrelevant, but this is relevant because when we start to talk about the gut microbiome and its impact or influence on mental health states like schizophrenia, bipolar disorder, and depression, there is also, there is emerging, quite promising data showing how changes in the gut microbiota affect mental health state and improvement in mood upon upon changing the gut microbiota through various different manipulation mechanisms, including phage therapy, including fecal transplantation. But what you're here to learn about is drug-induced liver injury, yeah? And this is some data taken from the UK Biobank, published by a colleague of mine in Germany, and looking at a huge number of patients. What we can see here is that long-term PPI intake. This is not acute drug-induced liver injury from a PPI. This is chronic PPI use over a number of years. PPI users have a significantly greater risk of developing acute liver failure. This is not just by taking the PPI. Their incidence of developing all-cause acute liver failure, not chronic liver disease, acute liver failure, is significantly greater with longer-term PPI intake. Now, okay, of course, the numbers are small. Acute liver failure is thankfully a rare condition. But we see a similar picture when we look at long-term antibiotic intake. Long-term antibiotic intake is associated with a heightened risk of acute liver failure. And you can see the odds ratios there at the bottom. Now, why does this happen? What are the potential mechanisms? Now, we understand from those, from these tables that I showed earlier on, that yes, PPIs, the antibiotics are going to be affecting the the way that the gut microbiome. But let's take a step back here and try to understand some of the mechanisms by which acute liver failure may be manifesting in this particular patient population. Oh, sorry, I think somebody just was unmuted. So let's look at models of steatohepatitis. Now, again, another a landmark a publication from about 10, 11 years ago now looked at the NRLP 6 inflammasome. This particular sensor detects the gut microbiome and is in part responsible for inducing immune tolerance. And when you disrupt this, this inflammasome, you not just weaken the intestinal barrier and drive translocation, but you also promote steatosis and inflammation because of that. And when we look at inflammasome knockdown, you can see significantly greater steatosis and hepatitis in animal models. Now, when we knock down the inflammasome in mouse models, we see a reduction in that very important bacterial diversity. We see reduction in certain species, such as Firmicutes, such as Lactospiraceae, and indeed Bacteroides, right at the very start before we even induce paracetamol or acetaminophen-induced liver injury. And when we look at these particular animals who have a knockdown, who are lacking this important inflammasome contribution, we see significantly greater a significantly greater burden of APAP-induced liver injury, acetaminophen-induced hepatocyte, hepatic necrosis, and much higher, ALT values, suggesting that this loss of this protective inflammasome sensor is heightening the immune response, heightening the this toxic liver injury, both pathways, not just one. But what happens thereafter if you perform adoptive transfer models of fecal transplantation? Well, as I mentioned, it is not that difficult in liver disease studies to induce disease. And certainly if you take wild type animals and animals who have this particular inflammasome and they've been given paracetamol induced liver injury, we can in fact transfer the risk of hepatic necrosis purely by transferring the gut microbiome from these animals. So you've got a knockdown animal of this NRLP inflammasome. You've given this animal paracetamol-induced liver injury, acetaminophen-induced liver injury, and you're able thereafter to induce the same histological phenotype in a wild type animal purely by transferring the gut microbiota without the additional risk of paracetamol. So certainly trying to modify the gut microbiome in a more favorable way, which I'll sort of come on to in a few slides, may be a therapeutic avenue in this particular disease, which is certainly the commonest cause of acute liver failure in the Western population, unlike viral hepatitis in the East and where I'm from. Now, I mean, sort of as hepatologists, it is not uncommon to liaise very closely with our oncologists who see a wealth of liver toxicity, and certainly they push their boundaries of toxicity more than any of our other professional colleagues. And cisplatin is not used that commonly these days, but it's certainly a well-recognized cause of liver injury. And in a similar vein to what we saw in the APAP models, when we look at cisplatin-induced liver injury, we see a lower degree of bacterial diversity and very distinct separation compared to what we see with control animals. Now, the reason that we one second, yeah. So this should say associated with rather than mediated by. The next slide is the mediated by enteric dysbiosis, because what we see here is that when we give antibiotic pre-treatment to cisplatin-induced liver injury, or give antibiotics very soon following cisplatin exposure, these animals do not develop liver injury. So this dysbiosis is preempted by the treatment of specific antibiotics in oral vancomycin, in oral metronidazole, which has been shown to mitigate liver injury in these animals. Now, I'm ashamed to admit I don't see enough cisplatin-induced liver injury or enough patients who have been before they are started on cisplatin to do a prospective trial of oral vancomycin or metronidazole before they receive their chemotherapy. But it would be interesting to see in this regard whether we can have similar observations in the human population. Now, when we again perform these adoptive transfer models, you can so we see the similar sort of picture as we saw in the paracetamol picture. We see transfer of fecal transplantation from these particular animals is associated with the induction of acute liver injury in a very similar way to cisplatin alone. So transferring this this gut microbiota to wild type animals, it reproduces that particular this particular phenotype. But let's move on slightly now, beyond just talking about the way that the gut microbiome is able to induce acute liver injury, and talk a little bit about bile acid metabolism. So one of the things that we've come to learn in the last 10, 20 years is that bile acids, rather than just being an important component of bile salts and in mitigating digestion, is the very important role they have as nuclear signaling molecules. We recognize for a while that they do mitigate to some degree symptoms such as pruritus, but more recently we've started to learn that they're able to shape and polarize critical immune-mediated responses, not just within the liver, but also within the gut, and also from the gut to the liver. There are particular pathways which I'm going to call out now. Now, two distinct derivatives of lithocolic acid, 3-oxolithocolic acid and iso-allolithocolic acid. These particular secondary bile acids have been shown to inhibit the differentiation of Th17 cells by directly binding to raw gamma T and promoting differentiation of regulatory T cells. They upregulate FOXP3, and all of this is mediated by the by enzymatic machinery, hydroxysteroid dehydrogenase. Now, the key bacterial producers of this are going to be your Actinobacter, your Bacteroides, and your Firmicutes, and these phyla are reduced in abundance in people who have immune-mediated liver diseases such as autoimmune hepatitis in PSC, and also reduce sort of in proportion in people who develop chronic liver injuries sort of related to compounds such as minocycline and nitrofurantoin. Now, many of you are aware about, as I touched upon right at the beginning, about the studies that we've done in PSC and inflammatory sort of bowel disease. And one common feature that we see in many forms of immune-mediated injury, including chronic immune-mediated hepatitis from the aforementioned drugs, is this reduction in bacteroides. And this is one of the main sort of players that is responsible for polarizing your Th17 to regulatory T cells. And when we talk about bile acid signaling, the key molecules, or the key receptors, should I say, which have been studied therapeutically in recent times are FXR, which is the rate-limiting step of bile acid synthesis, and to a lesser degree, PPAR, and also TGR5 or G protein, sort of a coupled bile acid receptor. Now, one thing that we've seen is that when we administer parenteral nutrition to our patients who have intestinal failure, then drug-induced, I wouldn't necessarily call it drug-induced liver injury, but intestinal failure-associated liver injury is a well-recognized picture. Now, historically, this was due to the component of the feed, which has now changed to have more olive oil rather than sort of unfavorable fats within the feed itself. However, we still do see parenteral associated cholestasis due to other things, for example, due to sepsis. And some of the histological features that we see in parenteral associated sort of liver injury, and I'll come on to talk about non-steroidal induced liver injury, can be attenuated following FXR activation. So when we activate FXR in parenteral nutrition-associated cholestatic sort of models in mice, we see almost complete abrogation of liver injury. And there are very nice papers in the bioinformatics domain showing that if you, which suggest that the toxic effects of NSAID-induced liver injury are because of antagonism of FXR, and because we have ongoing if we have ongoing antagonism of FXR, we have no cap, no break on the synthesis of toxic bile acids. So the toxicity of NSAIDs begins. FXR is antagonized by NSAIDs, and we don't switch off that particular break. So yes, I do. I do wonder whether molecules like Silofex like obeticholic acid, like Topifexor, are one of these particular models that molecules that we should study the attenuation of drug-induced liver injury, particularly because they have very high potency at the ileal FXR, not just in the hepatobiliary tree. But what about TGR5? Less sort of well studied, but we see similar things. When we delete TGR5, which remember sits upstream of the pathway of iso-lithocholic acid and 3-oxolithocholic acid, if you switch this cycle off, then we exacerbate acetaminophen-induced liver injury in animal models, suggesting that whilst we still believe that this is all NABQI related or toxic liver injury related, this particular immune bile acid break plays a critical role in the induction of liver injury. And reciprocally, when we activate TGR5, activate GBPAR, then using agonists either BAR or either CHIN117, we are able to reverse this particular pathway of liver injury from acetaminophen. Now, I'm not saying that we should be using obeticholic acid to treat our patients who've got acetaminophen-induced liver injury. That's not what I'm saying at all. Obeticholic acid does not work on TGR5, but it's just saying if we have particular mechanisms that enhance the production of isolithocholic acid and oxolithocholic acid, that may be able to mitigate some of the unfavorable immune responses which drive bad outcomes in these particular patients. When we see patients who have acetaminophen-induced liver injury, and we see people who in that particular group who don't go on to need things such as liver transplantation, then they get over that immediate liver failure danger zone. It's sepsis that kills them. It's that septic hit, that immune activation hit, which if we're able to mitigate somehow by sort of by lowering our Th17 responses, then that may be partially sort of favorable. Sorry, just slide stuck. And sort of lastly, I just want to talk about lymphocyte recruitment. Now, I mean, the vast majority of the liver's blood supply comes from the portal vein and sort of and passes through the sinusoids out into this the liver extracts sort of what it needs to, and through this particular process we see trafficking of lymphocytes, trafficking of mucosal lymphocytes, as well as those that are primed sort of within the portal lymph nodes. And these particular lymphocytes need to be able to home, be able to traffic to sort of where they need to go. And this, of course, is mitigated by a series of chemokine receptors, integrins, and their associated receptors. Now, it was long believed, including by sort of people sort of like myself, that the whole gut liver homing hypothesis was going to be something unique to immune-mediated liver injury. And whilst there are some unique principles behind it that I'm not going to go into, one of the things that we've started to see is that trafficking of alpha-4 beta-7 lymphocytes from the gut to the liver is a common phenomenon in sort of not just chronic but also acute forms of liver injury. And that makes sense, because in acute liver injury or chronic liver disease, when we get that bacterial translocation and we get heightened activation of the mucosal immune system, you want your mucosal immune cells who have seen those particular insults to traffic to the sinusoids. Now, I've used alpha-4 beta-7 and MAdCAM1 here as an example, but the expression of these adhesion molecules, MAdCAM1, and indeed other molecules such as VCAM and ICAM, is driven in part by the activation of certain amine oxidases. Now, vascular adhesion protein 1, VAP1, is something that's that is an adhesion molecule and an amine oxidase, and we all express it constitutively in our livers, but levels become upregulated during states of hepatic inflammation, particularly, and the factors that drive this are generally thought to be gut-derived amines, and particular amines released from the colonic epithelium, such as cysteamine, such as methylamine. And the amines alone are not going to be able to drive this upregulation of gut-specific adhesion molecules under states of stress. You need inflammatory signatures, and unsurprisingly, TNF is the most potent activator in that regard. Now, I'll come back to sort of TNF sort of in a second, but I just want to sort of put this up here, and hopefully everybody will get this this right. These are two parallel series of liver biopsy. One is checkpoint inhibitor-induced liver injury, and one is de novo autoimmune hepatitis. And I don't know if people want to vote in the chat just for a bit of fun, but I would just like people to tell me if they think the top one is autoimmune hepatitis, or if they think the top one is checkpoint-induced liver injury, and obviously the one below is going to be default. So if anybody wants, can people just put in the chat if they think the top one is de novo AIH? If people say, people can just say yes in the chat. Okay, if people think the top one is going to be checkpoint inhibitor-induced liver injury, can people say yes in the chat? I can't see anything in the chat, so I don't know what people are voting. Okay. Yeah, I can't see anyone in the chat, so I'll negate that. I'll put people out of their misery. But here you can see, obviously, checkpoint-induced liver injury looks very different to autoimmune hepatitis, and I've brought that to the attention because when we first started seeing and managing people with checkpoint inhibitor-induced liver injury, then we started managing it like autoimmune hepatitis. And whilst immunosuppression is still a cornerstone of managing this particular condition, some of the treatment algorithms do diverge. And one of the sort of earliest sort of agents that we used to treat checkpoint inhibitor-induced liver injury was anti-TNF. We used infliximab to treat this particular disease. And yes, if you are from Germany and if you've trained with Professor Loser and Christoph Schramm's team, they published some very nice data early on regarding checkpoint inhibitor-induced regarding anti-TNF agents treating autoimmune hepatitis. But of course, anti-TNF agents can cause or lead to drug-induced hepatitis. But in checkpoint-induced liver injury, the plot is slightly more difficult to dissect. What we see in people who, at least in animals who have checkpoint-induced liver injury, and certainly checkpoint-induced inhibitor-induced colitis, is that prophylactic treatment of using TNF uncouples this toxicity from efficacy. So you still get the efficacious actions that you desire from your checkpoint inhibitors in terms of mitigating tumor growth, but pre-treatment with infliximab seemingly almost knocks out the risk of developing colitis and also knocks out the risk of developing liver injury. Now, the reasons why this may be can be widely hypothesized and speculated. However, one of the reasons why we believe this, and with things that we're trying to prove in our lab, is that pre-treatment with infliximab actually reduces VAP1 mediated upregulation of particular adhesion molecules, not just to the gut but to the liver. And so trying to selectively identify whether you're attenuating mucosal injury in the gut first, even if you're not getting colitis, and to see whether that is able to mitigate liver injury, something that we're exploring. Okay, so that's just a very short summary of key points regarding mucosal immunity, mainly associated with risks in acute drug-induced liver injury. I've hopefully shown you some nice data from our colleagues in the Netherlands regarding how the gut microbiome becomes established in early life and the factors influencing its stability relating to cohabitation more so than genetic contributions. We see that loss of inflammasome sensing, at least in animal models, and not just associates with enteric dysbiosis, but seemingly augments, acute liver injury from acetaminophen, and certainly in the cisplatin models as well. I've illustrated some data regarding the protective impact of particular secondary bile acids and how loss of key bacterial components may lead to loss of production of lithocholic acid, oxalithocholic acid, and upregulation of, or should I say, a loss of polarization of regulatory T cells and more of a skew towards persistent Th17 phenotypes. And also how FXR agonism, at least in animal models, affects NSAID-induced liver injury, and in a similar vein, how TGR5 seemingly affects APAP-induced liver injury. And I do think the therapeutic potential of attenuating mucosal lymphocyte homing from the gut to the liver needs exploration in drug-induced liver injury, particularly when we are trying to understand the mechanisms of checkpoint inhibitor-induced hepatitis. Thank you for your time. Happy to take any questions. If there are any.
No, sorry, Palak, I could not activate my audio.
Yeah.
So thank you very much for this excellent talk. I really enjoy it. Since I'm aware of many of the papers that you showed, and specifically with inflammasome knockouts and on all these TGR5 is really extraordinary. Do we have any questions in the audience?
Yes.
Go ahead, please.
Hello, Palak, and congratulations. It's been really an excellent talk, very interesting. In the verge of the research in the field. My question is, with regard to the colitis induced by immune checkpoint inhibitors, and the attenuation of colitis, the mitigation by use of TNF antagonism. This could be probably also applied to the inhibitors, the immune checkpoint inhibitors induce liver injury, but the problem is we don't have yet a specific profile that are able to identify this patient. I would like to have some comment from you on this topic.
Yes, yeah, sure. So, as you know, you and I, we're part of this EASL registry grant for the checkpoint inhibitor. So we've started collecting cases, and there wasn't funding to collect stool from them or biobank specimens. That's something that we're trying to do in-house. But what I'm hoping is that if we're able to identify changes in the gut microbiome that precede development of liver injury, hence people who start on checkpoint inhibitor, any checkpoint inhibitor, of which there are hundreds and hundreds, thousands and thousands, collecting at a single time point their stool and their blood, and when they develop liver injury, collecting samples in a similar way. That sort of efficacy and mechanism evaluation is what we need to do. And if we're able to find out that these particular, just for sake of argument, if these particular patients have a reduced diversity as compared to those who don't develop injury, and in particular, they're not producing enough protective bile acids, then those particular individuals may be candidates for trials, at least, of prophylactic anti-TNF. I know there was a phase 1 trial that was planned. I don't think it's recruiting, though. I'm not part of that collaboration. From the group who did pre-treatment with anti-TNF. I think it was out in 2019, that paper. And the pandemic happened afterwards, so maybe there was some barriers, but that particular trial was specifically looking at the pre-treatment with infliximab. The final thing I'll say is, given that quite a high proportion develop it, relatively speaking, if you're on dual agents, then some of our oncologists report up to 30% of people develop some form of liver injury with it. It's almost worthwhile saying that developing a predictive model along the lines of which you're saying, not just from a basic science point of view, but even clinically, whether there are any other circulating bile acids that we can quantify more readily, which rather than even doing any sophisticated microbiome analysis that may predict who's going to develop and who's not going to develop it. So that's my 2 cents. Is I think we need to prospectively biobank, look at bile acid profiles as well as gut microbial profiles, and start with the lowest hanging fruit first.
Thank you very much. Very interesting. Congratulations again.
Gareth, go ahead if you have a question.
Yeah, hi. Nice talk. I have a question. Obviously, it's a follow-on really from what you've been saying about the TNF trial, and I've read a little bit into this. There was issues with the TNF trial because in the context of the liver, it's always under inflammatory insults per se. So obviously inflammation is probably a good thing in one sense for a healthy liver to function. But while there must be like a tipping point in which inflammation becomes an issue. So is there, in the context of your microbiome, does that sort of tally up in the sense that there's good inflammation, bad inflammation in terms of like good gut flora, bad gut flora? Yeah, bit naive question, but
no, not at all. I think I understand where you're coming from. So, yes, it'll be. I don't think it will be as simplistic as good and bad. It's more about the balance and the duration. So, the whole concept of pathobionts are that we all have enterococcus in our body. We all do. But it's when does that become dangerous? In what quantities does it become dangerous? What is the tipping point in function where it becomes dangerous? This is going to sound like a slightly ill thought through analogy, but when I was trying to teach, talking to my older son about what we were doing, then his analogy was, Anakin Skywalker in Star Wars was always Anakin Skywalker, but there was something that made him into Darth Vader. So the presence of those bacteria is always there. Similarly with the immune cells, these immune cells are always there, but it's when that inflammation goes unchecked for too long and when they start to develop characteristics that are unfavorable. That's what we need to sort of identify. And when you've got somebody who's got, say, a HCC, a liver cancer, you want to have active immune cells to target it. You want to get rid of that tolerogenic environment. But when you've got people who've got sort of active liver injury to the point where you're worried that they're going to compromise the synthetic function, and that's not allowing you to use a life-prolonging treatment, and these checkpoint inhibitors are great drugs; they prolong life for years, not just days, weeks, or months. That's the challenge. And if the answer does lie in the gut microbiome, and obviously I'm biased because that's my area of interest, then yes, I do think that we should be identifying bile acid signatures, gut microbial signatures, inflammasome signatures that predict who's going to get injury, because those are the people that we're going to want to pre-treat in such a trial.
And sorry, another quick question is, if you don't mind,
yeah, sure.
Obviously, my first degree was microbiology and virology, so medical microbiology. So it's quite interesting in a sense. Have you looked at, in the terms of obviously the metagenomic profiles of like the involvement of phage as well?
The short answer is no. I haven't. I can perhaps mention one thing, but I'm slightly bound by a CDA, so I can't mention everything. And obviously we're being recorded. But I know some phage trials in some elements of medicine have actually led to acute liver injury and have been terminated. Now, whether that is because of whether that's because the phage is actually causing direct liver injury or is actually leading to, I haven't talked anything about virome at all, the gut virome, but whether that's because we're unfavorably affecting that, I don't know, is a short answer. But there are some challenges with phage therapy, and some of the studies have not taken off because there is a liver injury risk.
Mm-hmm. So, Palak, I have one question there. So when you're, as you showed, what do you think is more important, the inflammasome activation? Is in the gut or in the liver? Because what you showed is, this paper from Schneider, no? It looks like it's a complete knockout mouse, no? So it's not a specific.
Yeah, it's true. We like to think of the liver as a sterile environment, right? We like to think about that, and what we would need to do to answer that particular question is use such a model, I don't know, like a Cree model or or have a targeted knockout, and those are being developed. They're not, and I'm sure sort of have been used in other circles at the moment.
Yeah.
But I think because it affects the gut microbiome to such a degree, I am inclined to think it's going to be more the gut that sort of causes the disease, and that's because the change in the gut microbiome occurs first, and in the steatotic models, the steatohepatitis occurs later.
Exactly. Yeah, I think so too. So how if you're trying to treat as Gareth just asked you, and the phages are not working properly, would you recommend fecal matter transplant as a source of to change the microbiota in acute liver injury, because in chronic?
Yes. So there are studies happening elsewhere in the UK to looking at acute on chronic liver injury. So sort of to answer that question, it's very challenging because of how sick acute liver failure patients are. But acute liver injury without failure, yes, I do think when they've got chronic disease, and it's whether you deplete with things like vancomycin first, like I showed you, versus before you give fecal transplant, and the route of fecal transplant. That's certainly something to try. And our experience of fecal transplant, we've always had to have a reason to give it to patients, but we've used it in a couple of people who've had nitrofurantoin-induced liver injury that wasn't resolving, and they got better. But the reason we gave fecal transplant was not because of nitrofurantoin, it's because the nitrofurantoin gave them Clostridium difficile, and we gave them fecal transplant because they weren't responding to. So how much of that was bad C. diff driving bad liver injury, and you've treated the C. diff and the liver got better? Who knows? But the short answer is yes, they are ACLF trials going on, but they're not specific to drug-induced liver injury, to my knowledge.
Exactly. Okay. Any other question from the audience? Yeah, Gary, go ahead again.
This one more. So no, it's interesting as well because you know, cisplatin. I seem to remember reading a paper many eons ago about cisplatin activating lysogenic E. coli and bacteria. So what is the role of like toll receptors in this?
Yeah. So that's a very difficult question to answer because I didn't really talk much about the sort of toll-like receptors at all. But I mean, I suppose it's not me skirting away from the question. I will come back to TLRs in a second. But the other thing about E. coli is that when they're active, they release lots of amines that activate amine oxidases within the liver. So one of the things that we're still looking at actually in the PSC field is why does the inflamed colonic epithelium and why does Escherichia, not just Escherichia coli, but other Escherichia groups, why do they release cysteamine when they're pushed? What is the mechanism of it? Is it and why does the colonic epithelium do it? Is it related to, there's another molecule called vanillin-1 I haven't mentioned, is it related to that? And why does that drive the upregulation of adhesion molecules within the liver? So we are looking at sort of the roles of sort of Escherichia, and it's a very good question about cisplatin and E. coli and whether that's another pathway. But I mean, of course, in any form of bacterial translocation, any leaky gut hypothesis, no matter what realm of hepatology you look like, then you see heightened toll-like receptor activation, you see heightened responses to pathogen stimulation, you see increased sort of PAMP and DAMP signaling. All of these things are there. I don't think any of these things will be disease specific, but in your case, in your question, cisplatin is the instigator, whether that's instigator by another insult, alcohol, or something, who knows? So I don't think it'll be disease specific, but it's a good question.
I agree. Any other question? Maribel, go ahead, please. Your audio, just turn on the microphone.
Hello, Palak. Thank you so much for your very nice presentation and very provoking presentation. You got a lot of questions. And I was going to ask you, because we've been trying to explore the microbiota in drug-induced liver injury, but as you have mentioned, drugs have an influence the microbiota, just only because taking drugs. And people who developed a drug-induced liver injury, you know that the main causes are antibiotics. And antibiotics, we know that they produce a disruption of all the microbiota. So, and apart from that, they may have other drugs being taken together. So how could you interpret or consider the differences that we get, for example, a diminishment in the diversity?
Yeah, no, it's true. So I suppose, I mean, I'll answer in a similar way to I think sort of Raul's question about the checkpoint inhibitors one. My thought process would be very similar. I mean, it's a dirty field, looking at microbiota, particularly given all the confounding factors and diet as well. I think that in order to do this, you would have to go for the lowest hanging fruit, your purest population of patients that you can have. And if we have very common, or not common, that's the wrong choice of words, if we have very well recognized cohorts of people who have very well recognized causes of drug-induced liver injury, like nitrofurantoin or even checkpoint inhibitor-induced injury, we have to say, right, we have to bite the bullet, and before they start a particular drug, you take a very, almost like we do with the UK Biobank or any other national biobank, you sample their stool and you sample their blood, and even urine, in fact, and right from the beginning. And when they get injury, you resample at that particular moment. And people who don't get injury at the similar sort of time point, you sample again, because then and only then will you be able to control for such factors.
Yeah.
In the UK Biobank study that I was showing, which was several 100,000 people. That UK Biobank has been going on for such a long time, and there will be many confounders beyond omeprazole, beyond other PPIs and antibiotics. But that's the only way that we can. And the other thing is just registries. Collecting every single medication that people are on, like we do in a trial. That's what Pro-Urodili, and so on are all about.
Yes, yes. But I think with the data that you have shown that mucosal permeability and the gut immunity may play an important role in the development of drug-induced liver injury. Yes. Bile acids, have been already shown as promising biomarkers in drug-induced liver injury. So also the connection that you have shown is very interesting.
Yeah.
Okay.
Thank you.
So thank you, Palak. It was an extraordinary talk today, and I just want to let you know that the next seminar will be the 22nd of February at 4:30, and it will be Paul Watkins who will be talking. So I hope that you attend, and I see you then, and I wish you the best until then.
Thank you.
Thank you to everyone. See you soon.
Thank you. Thank you,
Bye.
Palak. Thank you again. Thank you.